Good morning to all of you. Myself Dr. Ankita Singh. Today we are given about lecture about the haploid plants. Haploid defined as a sporophyte plant that contain a gametic chromosome number and number of chromosome, monoploid and polyploid. Haploid plant is characterized by possessing single set of chromosome in the sporophyte. This is contrast haploid which contain two set of chromosome. Haploid plants are great significance for the production of homogeneous lines. There is two type of haploids plant, monoploids and polyploids. Monoploids, these are haploid possess half number of chromosome from the diploid seed, and the haploid possess half the number of chromosome of polyploid species. In vivo and in vitro approaches. The importance of haploid, the field of plant breeding and genetic was realized long ago. Their practical application, however, has been restricted due to very low frequency. There is in vivo techniques, two types of techniques, androgenesis and gynogenesis. Androgenesis is male reproduction part and gynogenesis is the female reproduction part. Development of egg cell contain male nucleus of is to haploid referred to androgenesis. For a successful in vivo androgenesis, the age nucleus has to be inactivated for eliminated before fertilization. Gynogenesis, unfertilized egg can be manipulated to develop into haploid plant. Distinct hybridization. Hybrid can be produced by elimination of one of the parent chromosome and as a result distinct hybridization. Irradiation effect. Ultraviolet rays, X-rays are inactive the male part, either female part. Chemical dependence. Some chemicals are colchicine and other chemicals include chromosome elimination, somatic cell, which may result in haploids. In vitro techniques for haploid production, androgenesis and gynogenesis. Androgenesis is the male gametophyte produce haploid plant. The basic principle to stop the development of pollen cell into gamete is forced to develop haploid plant. There is two type of approaches, anther culture or pollen culture. Anther, the selected flower buds of young plants are surface sterilized and anther removed along with their filament, and anther are excised under aseptic condition and crushed in 1% acetomarcarpine. That takes the stage of pollen development. If they are not corrected stage, each anther is genetically separated and the intact anther inoculated on a nutrient medium. Insert anther should not be used in culture as they result in calcium of anther cell tissues. Pollen culture. Haploids can be produced from the immature pollen or microscope. The pollens be extracted pressing the squeezing the anther of the glass rod against the side of breaker. The pollen suspended in a filter to remove the anther tissues and debris. Comparison between anther and pollen. Anther culture is easy and quick participle. Anther walls are conditioning factor promote culture growth. Thus, the anther culture is responsible efficient haploid production. The major limitation is the plants not only originate for pollen but also from the parts of anther. Development of androgenesis haploids. The process of androgenesis ultimate production of the haploid plant is displayed. In this video, we can understand how the androgenesis plant will be developed. Gynogenesis. Gynogenesis is the in vitro culture of unpollinated ovaries and usually implied. We select eggs or we cut the eggs, unfertilized flower. This flower will be stored in 24 to 48 hours. Anthesis from the pollinated ovaries. Calyx and corolla stems will be removed and this ovary will be cultured in a medium. Limitation practice production of haploid plants ovary culture is not used too frequently as anther pollen culture in crop improvement. The dissection and unfertilized ovary ovules is rather is difficult. Thank you. Good morning to all of you. My name is Dr. Ankita Singh. Here I am presenting haploid culture in plants. Introduction. Haploids defined as the sporophyte plants that contain a gamete chromosome number n, which plant have an n number of chromosome, that is called haploid culture. Monoploid, just for example, monoploid n x is equal to 7. The gross plant is a haploid plant. Haploid plants are characterized by possessing only a single set of chromosomes who have a n number of chromosomes that are called haploid plant in which sporophyte. This is a contrast to diploid which contains two sets of a chromosome, that is called diploid culture. Haploid plants are great significance for the producing homogeneous line for the improvement plant and breeding program. Grouping of haploids may be divided into two broad categories, monoploid and polyploid. These are two haploids that possess half the number of chromosomes form of diploid species. Maize and barley. The haploid possesses half the number of chromosome polyploid species regardless polyploid wheat potato. It may be noted that the term polyploid generally used and applies to any plant organization for sporophyte it containing half number of chromosome. It means male have a half number chromosome and female have a half number of chromosome. In vivo and in vitro approaches. The importance of haploid in the field of plant breeding, the genetic was realized long ago. The practical application, however, is restricted due to very low frequency. The process of apomixis pathogenesis to responsible for the spontaneous natural production of haploid. Many attempts were made both in vivo and in vitro method. In vivo and in vitro method. In vivo and in vitro method in the wild method. In vivo techniques for haploid production, androgenesis and gynogenesis. Androgenesis is called male productive part and gynogenesis is a female productive part. Androgenesis, development of egg cell containing male nucleus of a haploid refers. Androgenesis, for a successful in vivo androgenesis, an egg nucleus has to be intactly eliminated before fertilization. Gynogenesis, egg part, the oval part, manipulated by and developed into haploid plant. And third is distinct hybridization. Hybrid can be produced by elimination of one of the parent. One of the parent means there is a male parent will be eliminated, either female parent will be eliminated. Irradiation effects. Ultraviolet rays. Exposure of ultraviolet rays is called irradiation effects. Chromosomal breakage and their subsequent elimination to produce haploid. Chemical treatment. Certain chemicals like maleic, nitrous oxide, colchicine, it will be helpful for the making haploid. This chemical treatment inactivates the one male part, either female part. In vitro techniques for haploid production is androgenesis and gynogenesis. Androgenesis, the male gametophyte produce haploid plant. The basic principle is stop the development of pollen cell into gametes and force to develop into haploid plant. There are two approaches, androgenesis, anther culture or pollen culture. Only the plant have n number of chromosomes in pollen or in egg. Anther culture. Selected flower buds of young flower, surface sterilized and anther removed along with their filament. These anther are exceed under specific conditions and trust them in a 1% acetocarmine and death stage of pollen development. In their correct stage, each anther is genetically separated and intact anther included on the nutrient media. Artificial nutrient media which are made from the MS media. The anther culture are maintained in alternating periods of light and darkness, 28 degrees centigrade. As the anther proliferation, they produce callus which later form an embryo that is called haploid point. Pollen culture. Haploid point can be produced from immature pollen or microspores. The pollen can be extracted possessing the sequencing the anther with a glass rod against the side of beaker. The pollen suspension is filtered to remove another tissue debris. Viable and large pollens are concentrated by filtration, washed and collected. These pollens are cultured in solid or liquid media, MS media. This is called haploid plant. Comparison between anther and pollen. Anther culture is easy, quick and participle. Anther walls act as conditioning factor and promote culture growth. Development of androgenic haploids. The process in vitro androgenesis of the ultimate production haploid plant is dispatched. In this diagram, we can understand how the mitosis and meiosis will occur and how the n number of chromosomes, we can say haploid plant can be made. Treatment methods are variable and largely depends on the donor plant species. Chemical treatment, temperature influence. Temperature will be influenced to the effect of light, effect of culture medium. Gynogenesis. Haploid can be developed from the gynogenesis, from the ovule, from the egg. In vitro culture, unpollinated ovaries. Which ovaries are not pollinated, not fertilized, this ovary will be taken and Flower buds are at least 24 to 48 hours prior to anthysis from the unpollinated ovaries. After removal, calyx, corolla, stamen, ovaries are subject to surface sterilization. The ovary with a cut and distal part of pedicle or solid culture media. The stigma, corolla, style, and cesium ovule. This ovule will be cut. When there is liquid media used, the ovaries are placed on the filter paper, allowed to float over the medium with a pedicle instead through the filter paper. The commonly used media is MS media, wide and 6 media. In this media, we grow the ovary. Limitation. In practice, production of haploid can be ovary and ovule culture is not used frequently as anther pollen culture is crop improvement program. The major limitation of the gynogenesis, the dissection unfertilized ovaries or ovule is rather difficult. The presence only for one ovary per flower is another disadvantage. Okay, thank you.